So, no, that's not what I want. I want the chat. Okay. And here we go. So just like with carbons and alcohols, we can classify amines, okay? Here we're looking at the carbon bonded to the amine group, okay? An amine group is this. It is some kind of carbon bonded to some kind of nitrogen group. Now we know how many bonds can nitrogen have in a compound?
Is it 3?
3, and it has a lone pair. So it's got one bond that you'll always see here to carbon, usually a single bond. It could be a double bond. Usually it's a single bond. So we've got 2 other places this nitrogen can have bonds, and then that nitrogen always has a lone pair, okay? So these other places, these could be bonds to other carbons, or these could be bonds to hydrogen, or any other atom, but a lot of the times, mostly it's carbon and hydrogen. So this is why, depending on what these other bonds are, how we classify amines. So an amine is a compound that has 1 or more organic groups bonded to nitrogen. And when I say an organic group, that's a vague word. It could be carbon, it could be hydrogen, it could be oxygen, it could be several things. So we just say organic groups. A lot of times any carbon groups we call alkyl groups, okay, like a methyl group or an ethyl group or a propyl group. So it could be a variety of things. So if it's a primary alcohol, you have 1 bond to carbon. So this R stands for a carbon group here. And then you have 2 bonds to hydrogen. So we represent it as RNH2. And that looks a whole lot like this right here. We call that a primary amine. Okay, and you still see the lone pair on nitrogen. A secondary amine is a nitrogen bonded to 2 carbons. So we have a subscript of 2 for the R now. And it only has 1 hydrogen. Okay, that's this picture right over here. You see the 2 carbons highlighted and then just 1 bond to hydrogen. That's a secondary amine. And you still got the lone pair. Moving forward, tertiary amine, here's what I noticed. Here's what's different. You have no hydrogens on this carbon now. All 3 bonds to nitrogen are occupied by a carbon, okay? That's why it's R3, 3 carbons. R stands for the carbon groups. So it's going to look something like this over here. We call that a tertiary amine, okay? Notice, though, nitrogen always has lone pairs, okay? Now, you can also get nitrogen in a ring structure. You can have a cyclic amine, or you could have, well, this is actually a special kind of group. I'm going to jump over this one right now. We'll recognize some specific nitrogen ring structures later, but you can have nitrogen in a ring structure. Just know that. We call them heterocycles, and we'll get to those in a minute. Now, the only time nitrogen kind of disobeys the rules is when it becomes what we call a quaternary amine. Okay, so upon the binding of a 4th group to nitrogen, which happens through the lone pair, that's the only available spot left. That lone pair can become a bond. We call this a quaternary. Now we don't just call it an amine, we call it has a different name because it becomes the charged cation. So we call it a quaternary ammonium cation or ion. All right, so that's when you've got 4 different carbon groups, 1, 2, 3, 4, all bonded to nitrogen. And now you notice that there is a plus charge on this nitrogen, okay? So technically, usually, well, I'm not going to get into the electrons of it, or the electron stuff and why it has a plus charge, but it does. And here's just an example. We have all these different ethyl groups bonded to this nitrogen down here, got a plus charge. So often when this happens, you will see nitrogen in its own sort of like ionic compound. You guys remember ionic compounds are made up of cations and anions? Here we have this giant cation, and the positive charge is right here on the nitrogen, and then we have the anion. It's a chloride anion. So we're gonna, anytime we talk about nitrogen as an ammonium ion, it's usually in an ionic compound, okay? Or it's usually there's some anion associated with it, and we'll name the anion with it. Okay, now all of these are derivatives, and when I say derivatives, they're like different forms of the most original amine. The most original amine you can have is up here, N with 3 H's. Anybody know what this compound's called? It's actually a very important compound when it comes to agriculture and industry. It's used in fertilizers a lot. We make tons of it industrially. It's called ammonia. And it doesn't smell good. Okay, ammonia really doesn't smell that good. Okay, so the reason that we say this is a quaternary, I'm going to highlight it, ammonium ion is it's a derivative of ammonia. All of these amine structures are derivatives of this original simplified ammonia right up here. Okay. Here's just some examples. I just threw this figure on here to show you some examples. So this methylamine, all right, it's a primary. We can call them either alkyl amines or aryl amines, okay? If it's got a CH3 or an ethyl or some kind of straight chain group branched off of it, those are alkyl groups branched off the amine. So you might hear the phrase alkyl amine. That's if it has straight or even branched chains coming off the amine. But if it has an aromatic group attached, we'll call them aryl amines, A-R-Y-L. Okay, so it's the difference between alkyl and aryl. All right, or you might see it say aromatic amine, aryl aromatic, kind of the same thing. So those are just different examples of how it might look in real life. Okay, let's go on to how do we name these and how do we draw these. So again, there's going to be some slightly different rules depending on if it's a primary, a secondary, or a tertiary. So if it's a primary, meaning you just have one alkyl group bonded to your nitrogen, all you do is you identify the alkyl group, you name it, and then you add the suffix, meaning it comes at the end. It's not a prefix. Prefix comes before, suffix comes afterwards. You add the suffix amine. So for example, look at this first one. I'm going to zoom in. You have this ethyl group here. How do I know it's ethyl? It's a 2-carbon substituent. So a 1-carbon substituent is meth, then eth, then prop, then but. You know these words by now. So we just call it ethyl, and then the suffix is amine, ethylamine, all one word. Okay. We don't have to name the hydrogens or really acknowledge them. Okay. Now we have a different group. We have a 3-carbon group, and I noticed that the amine is bonded through that center internal carbon. So this is isopropyl. So we just call it isopropylamine. If we keep going, now off of this nitrogen, we have a whole cyclic alkane, okay? And so we call it cyclo. There's 6 carbons, 1, 2, 3, 4, 5, 6. I probably didn't even number those the right way. I probably should have started with number 1 on the carbon branch to the nitrogen, but that's okay. There's 6 carbons in this. So we call it cyclohexyl. Notice the YL. This is kind of different. We actually haven't seen this exactly like this before. Cyclohexyl, then you have the suffix amine. Okay. So we don't say cyclohexane amine. We name it with a YL ending, just like we do any other alkyl group. Cyclohexylamine.
On the previous slide, why is it named like that? The primary aromatic amine.
Aniline?
Like I feel like all of them have the same thing. Yeah, I feel like all of them kind of have the same thing going.
Yes.
And then
that's a great question, Maggie. So remember there's 2 styles of naming, the IUPAC. This is the very systematic one. This is the numbers and the commas and the hyphens and lots of rules. And you're going to see the name structures look similar. Then there's the common name. And this is the part of organic I told you guys that frustrates me the most, because you can learn all the rules and all the systems, and then yet there's another name for this compound. And that's what you're seeing with aniline. We actually introduced aniline when we were talking about benzene. Okay, this is a benzene ring. Certain benzenes with specific groups branched off of them have specific names. That's why you see the word like phenol sometimes. Phenol is nothing more than a benzene with what branched off of it? Anybody?
Is it alcohol?
An OH, okay? But this has a common name, and the common name is phenol. It's the same idea here with aniline. Aniline is the common, what happened? Aniline is the common name for a benzene ring with an NH2 group branched off, okay? Another common one is if you have a benzene with a CH3. Anybody remember the common name for that? It's really hard to pronounce. Toluene. Okay, anyways, this is kind of besides the point, but this is exactly what you're seeing, Maggie. It's just this is the common. This is the common name. Okay. All right. Okay, we're going to do some practice with naming in a minute, but let me explain the rules for secondary and tertiary amines. When you have a secondary and tertiary amine, okay, when the attached groups are identical, you use prefixes such as di or tri or tetra, and then you also add the suffix amine. For example, look at this amine right here. Here's the lone pair on this nitrogen. We got one hydrogen attached and we have two alkyl groups. If you look at those alkyl groups on the right and on the left, they're identical. They both have 3 carbons. Okay, so these are propyl groups. So we just call this dipropylamine. If you look at the next one over, this is a tertiary amine. So this one, by the way, this is a secondary amine. This one's a tertiary amine. We have 3 identical groups. They're all ethyl groups. That's an ethyl group. That's an ethyl group. That's an ethyl group. So we call this triethylamine. Okay, now I know you're thinking, well, what if they're not the same? Well, when the attached groups are different, the rule is a little more specific. You're going to name the compound. Let me zoom out. Name the compound according to the largest attached group, the one that has the most carbons. And then you have to somehow acknowledge that there's other groups as well, and you have to name those. And you will, but you're going to use the prefix in to name the other substituents. Here's what I mean. Let's look at an example. Okay, you got this nitrogen right here. We have an ethyl group and we have a propyl group attached. Which one's bigger? Which one has more carbons? Cameron said it, propyl. So we're going to name the compound according to this. We're going to call it propylamine. So I would encourage you, write that down first. Then you're going to go in front of that and you're going to name the other substituent. It's an ethyl group. It is bonded directly to the nitrogen. So we're going to call it in to indicate it's bonded directly to the nitrogen. In ethyl, and you put a dash between the in and the actual name of the alkyl group. In ethyl propylamine. And the in ethyl part lets you know that, again, it's attached directly to the nitrogen. It's another substituent, but we have to name it according to the largest attached group. Okay, what about this other one? Another example. Another propylamine. This is propylamine right here, but we have to acknowledge these methyl substituents. They're attached directly to nitrogen. So notice, just the way we do with numbers, every attached group gets its own number. So if we had like 2 methyl groups bonded to like a cycloalkane, we would say 2,2-methyl. Okay, here we have 2 methyl groups bonded to the nitrogen, so we say in dimethyl. Okay, both of those methyl groups attached directly to nitrogen, so we say in dimethyl propylamine. Questions on that? I think we've got a practice problem in a little bit. We'll name some. This is for sure not all the examples that there are out there, but this is a couple that hopefully drives home some of these rules. For more practice on that, you can always do those back of the chapter textbook problems, the even ones. I have some on your syllabus. Okay. Now, what we've been doing is we've been naming a compound according to naming it as an amine. Well, there are times where there is an amine group on a molecule or on a compound, but it's not the only functional group. Sometimes the amine itself is the substituent that we name as like a prefix, and we don't name it as an amine compound. Okay, so in that case, we acknowledge the nitrogen is present in the compound, but we acknowledge it as an amino group. Now it's like the attached substituent on, and we got to name the main compound. Okay, so when that's the case, and it's an N2 functional group, we call this an amino group. Okay, when this is branched off of a compound, we have to acknowledge it as an amino group. So for example, the IUPAC name for aniline, okay? So we have this branched NH2 group. Now there is this common name aniline, but we can name it systematically with the IUPAC naming. We just have to name it like this, amino benzene. Okay, and we don't even have to put a 1 there because all the carbons are identical in the ring. We just know if it's the only substituent. If there was another substituent, then we would have to use numbers and things. But we're acknowledging this amino group, and we're acknowledging that it's attached on benzene, and it's the branch substituent here. The common name for this just happens to be aniline. So you see that one more often. Okay, so when this group is present in a compound, amino is used as a prefix. The exception is when aromatic amines are attached as a functional group. They are known by the common historical names. Okay, again, I've been using this prime example of aniline. So let's look at an example of that. I have this long carbon chain right here. It's an alkene because we have this double bond. So this is butene. Specifically, it's one butene because it's occurring between carbon 1 and carbon 2. We always give the carbons in the double bond the lowest possible index number. So here, this is carbon 1 and 2. There's carbon 3 and 4. So we're going to call this 3-amino 1-butene. We're acknowledging there's an amino group, NH2 group, branched off carbon 3. This one over here on the far right is interesting. You've got this whole aromatic compound plus you have a methyl group branched on the nitrogen. So we see that this right here is aniline. You could call it aminobenzene. That's the systematic name, but we're going to they use the common names a lot for this. And then we say the methyl group is branched directly off this nitrogen. So we say N-methyl aniline. Okay. So again, those are just some examples to illustrate these rules. The hardest part with naming for this is deciding, okay, should I name it as an amine, or do I name the compound and acknowledge that there's an amino group attached? It depends if there's other functional groups present. If the amine group is the only functional group present, you name it as an amine. So something, something, amine. If there's other functional groups present, such as a double bond, you name that compound by that functional group and you acknowledge the amino group. Okay? And remember, an amino group is just an NH2. So if you have NH2 branched off of something, then you acknowledge the amino substituent, but you're naming the compound as something else. Does that make sense? Okay. So let's put this into practice. Write the structure of N,N-diethyl-butylamine and identify it as a primary, secondary, tertiary amine. N,N-diethyl-butylamine. So the word that's connected with amine, butylamine, this is the largest substituent on the amine group. These others, we got di means 2 ethyl groups, also branched off because it's N, branched off the nitrogen. All 3 things are branched off the nitrogen. You have a nitrogen in the middle with a butyl group branched off, and then 2 ethyl groups also branched off the same nitrogen. So I'm going to start drawing it too. Here's my butyl group. Okay, 4 carbons there. And then I have diethyl, CH2, CH3. You can put the bond there if you want. Another C. People write this different ways. People will write it CH2, or they'll write the carbon first and the hydrogens after. So you see me write the methyl groups like this a lot. I hope that doesn't confuse anybody the way I wrote the hydrogens in. Okay, so this is an ethyl group. This is an ethyl group. They're both bonded directly to nitrogen. This right here is the butyl group. And we still have the lone pair. So is this a primary, secondary, or tertiary amine? Yeah, Robin said it, tertiary. Or so, yeah, tertiary. Okay, that is a tertiary amine. All right, if you're done with that one, move on to the next one. If you're still working on this first one, that's okay. The next problem says name the following compound, and then is it primary, secondary, or tertiary amine? So we saw an example of this earlier. This is not aniline because that ring is not aromatic. It can only be aniline if it's an aromatic ring. This is not aromatic. This is just a cycloalkane. We saw an example of this on the slides earlier. So if you're unsure how to name it, go look at one of those examples and you'll get a really good clue. Anybody think they have the name for it?
I don't have the name, but I have a question.
Yeah, Abby.
Only aromatic if what? I was
It-
gonna write that note down.
It is only listed as aniline if this ring right here is aromatic.
Oh, only aniline. Okay,
Notice
awesome.
we have a similar thing going on. We have a cyclic structure bonded to a nitrogen, but our ring down here is not aromatic.
Okay, thank you.
Yeah. So this is not aniline.
Would it be, this is probably wrong, but methyl cycloamino?
Robin, that's close. There's a few things we need to tweak on that. We do have a methyl group, so I'm going to write methyl because that part was right. What do we need to tweak about this though?
Do we need an N methyl or N dash?
Yeah, N-methyl because it's bonded directly to the nitrogen and it's not the largest carbon group. So it's a substituent. So we got to say N-methyl.
Okay.
Okay, what else do we need to change here? How can we finish this out? What is this? How are we going to name this part?
Would that be a cyclohexyl?
Yes, ma'am. So we saw this earlier. It's kind of weird. This threw me off too when I was looking at this example. Okay, this is what we call it is a substituent in this case, but it's the largest one. So we're going to name it like, it's kind of like the parent substituent in this case. So it's a cyclohexane, but it's a group branched off of the amine group. And the amine here is the only functional group present beyond an alkane. So we're going to name this with a YL ending. So it's N-methyl cyclohexylamine. So we saw this. Scroll up. We saw it right here. Okay. I think I have a poll question for this next one. Everybody got the name of this written down? Any questions on that? Okay, I want you to classify these. I'm gonna launch a poll. Okay. So look at A, B, C, D. I only have D. I didn't do E. Whoops. I put in my own question in place of E. So you're just classifying A, B, C, D here. Draw it out if it helps you. This is what I have for A, if that helps you classify it. For B, And then these would all be hydrogens. This is what I have for B. Okay, I'm most interested in the last question, in case you're wondering.
Do you want us to answer E too? It wasn't on the poll.
Yeah, I forgot to put it in on the poll.
Okay.
Yeah, go ahead and do E on your paper. Okay, we're waiting for 2 more votes. Okay, get your vote in if you can. We'll walk through all these, so even if you're not sure. Okay, I'm gonna end it. Share results. So everybody said on this, most people said on this first one, it's a primary mean. Okay, so we have this nitrogen is only bonded to 1 carbon. So that is a primary mean. These other bonds to hydrogen, we don't consider those when we're trying to classify this. I mean, we do consider them, but we're really only counting the nitrogen carbon bonds. So this is a primary mean. It does have 3 bonds. Nitrogen will always have 3 bonds unless it's a quaternary mean, but we're counting the carbon nitrogen bonds. Okay, number 2, B. This has 1 bond to carbon there and 1 bond to carbon there. So we do classify it as a secondary mean. It's got 2 nitrogen carbon bonds to different carbons. Now, what if you had something like this? Sorry, I just thought of a random question. What if you had this, a nitrogen double bonded to a carbon and then bonded to a carbon on this side? How would you classify that?
Would it be a tertiary? Because it has 3 bonds.
So yeah, the question is this secondary or tertiary? There's only 2 carbons here, but you're counting bonds. Remember, that's 1, this is another one, and that's a third nitrogen carbon bond. So that would be tertiary amine. So you're not counting the number of carbons it's bonded to, but the number of nitrogen carbon bonds. Does that make sense? The actual dashes, count the dashes. Okay, let's keep going. On C, most people said it is a tertiary amine. Okay, so we're looking, oh, sorry, this is C. So we've got that bond, whoops, wrong. We've got this right here. We've got the nitrogen bonded to 1 carbon. I almost did the same thing that a lot of y'all did. This is a primary. The nitrogen is just bonded to 1 carbon. Now that carbon, this carbon is a tertiary carbon, but I'm asking you to tell me, is this a tertiary or a primary amine? So you're looking at the nitrogen here. So the nitrogen, it's a primary amine. That carbon is a tertiary carbon, but the nitrogen specifically is only bonded to 1 carbon. That's why it's primary. D, most people said secondary. And if I highlight these bonds, we got a nitrogen carbon bond there, and we have a nitrogen carbon bond there as well. I don't know who that was. Sorry, everybody. So that is secondary. Okay, there's 2 nitrogen carbon bonds. Okay, so most people said that. Okay, E, we can go ahead and look at E. What do y'all think E is?
Is it tertiary?
Yes, it is. We got a nitrogen, whoops, I meant to highlight. 1 nitrogen carbon bond there, 2, and then 1 more right here. So this is tertiary, for sure. Okay, and the best ice cream in Conway is Andy's. Okay, I've never had Freddy's, and I'm really not a big fan of Marble Slab, I'll be honest. Nobody said store-bought. Robin, where do you go for ice cream?
I go to Freddy's. Well,
Yeah,
we
you
have
like
a Freddy's. We have a Freddy's. Sorry, my camera's off. I just think it helps with my hot spot. I don't know.
That's fine.
But we have Freddy's in Bryant, and I really like it because you can you can make it just like Andy's. You can kind of put whatever you want in it,
Yeah.
but I just like the ice cream better than Andy's ice cream. I can taste the difference in my ice creams.
Wow. So I love ice cream. It's like my favorite if I'm going to pick a dessert. My favorite ice cream is actually, you can't get it in Arkansas. How sad is that? It shouldn't be my favorite then. Has anybody heard of Jenny's? Jenis. The closest Jenny's store is in Nashville, Tennessee. That's the closest store. You could order it online, I guess. It'd be very expensive. You can sometimes find their pints in like Whole Foods or but it's kind of expensive.
Is it like in downtown Nashville? I go to Nashville a lot because one of my best friend's boyfriends live there.
Uh-huh.
So it's like
there's 3 Jenny's locations in Nashville. So it's like there's one in the there's a specific area that has, I forget the little districts, but there's 3 Jenny's right in downtown Nashville. Yeah. So anyways, nobody poor Sonic. I'm still a fan of Sonic. I go to Sonic sometimes. Just for their ice cream though. Okay. What do you get if you go to Andy's, people? Nobody gonna share their ice cream order?
I can't remember what I get. I know it's like strawberries and like banana and vanilla ice cream. I think it's
I
called like
like
the Strawyanna or something like that.
Strawyanna. That's
Mhm.
interesting. I do like their strawberry concrete. I'm a vanilla person. I don't get chocolate.
Oh, me too.
I get
Okay.
the one with the chocolate.
What?
I get the one with the what's the melted chocolate called, y'all? I always forget.
The hot fudge.
The hot fudge, yeah.
Yeah.
I get the one like with the hot fudge in the middle, and it's like vanilla ice cream all around, and I get strawberries and cheesecake. I always get strawberries and cheesecake in mine.
Oh my gosh. That sounds so good. My daughter doesn't care what flavor ice cream she wants. She only cares about the color of the ice cream. She just wants pink ice cream. So
That's the way I am.
okay,
My
Abby.
birthday cake.
That's how my 2-year-old is.
It's gotta be blue birthday cake.
Oh my. Wow. That's why you go to Marble Slab. Okay. Properties of amines. I could talk about food all day, in case you're wondering. So when we talk about properties by now, y'all probably grasp the fact that we're talking about boiling points. We're talking about, if it's a solid or a liquid or a gas at room temperature. Sometimes we mention that. We're talking about solubility. Is it soluble in polar things or non-polar things? Now, why would it be important that we're concerned about whether compounds with certain functional groups are soluble in water or not? Why do we care about this from a physiological standpoint? Any thoughts on why this is important? Think about the body. Nothing. No guesses.
Are we talking about why nitrogen is important? I'm sorry.
I'm talking about why do we talk about properties, period? Like, why do we care if molecules are soluble in water or not?
I was gonna say if we're talking about nitrogens, because that's a part of proteins and amino acids. So like the structure of compounds determine what the function is.
Yes, and we are headed there very soon. We're going to get to proteins next week. Because think, the body is mostly water, right? So if compounds and these compounds come to us in the form of our food, in prescription drugs that we take, in things that we drink, and so it matters if these things are soluble or not, because as we put things in the body is an aqueous environment. The body is a water-based environment. Okay. Now you do have non-polar areas, and those are like cell membranes, and we're going to get to all that. But these compounds, if it's water soluble, they will be soluble in the body. If it's not water soluble, so like say we're designing a drug, okay, people are coming up with new drugs all the time, and they're putting them through clinical trials. In drug design especially, they have to consider water solubility. Okay, for that drug to go into the body, it's got to be water soluble, or they're going to have to do a whole lot of extra stuff to get it in the body and where it needs to go. Does that make sense? That's why we care about water solubility. I wanted to just point that purpose out because we do talk about this a lot, and I don't think I had explained that yet. So let's consider amines. Before we just get to all the properties, I'm going to explain a few things. You guys, it's been a long time since we've done chapter 10, but we talked about acids and bases in chapter 10, and we said any molecule with a lone pair can act as a base. Okay, I nickname them in my class. I say they're proton acceptors. And when I say a proton, here's what I mean. I just mean a hydrogen atom that's been stripped of its electron. So look at hydrogen real quick. Hydrogen, if I draw, actually I'm going to draw a plus sign. There's the nucleus with the proton, and then here's the electron in the electron cloud, right? That's what an atom looks like. We know that. This is hydrogen, one proton, one electron. When we take the electron away from hydrogen, guess what? All that's left is just the proton in the nucleus. So when I say a proton acceptor, I mean this acceptor, an H plus acceptor. A hydrogen ion is just a proton. So we just nickname it a proton, okay? So any molecule with a lone pair can act as a base, and it can accept or make bonds with an H plus ion, okay? So look at this picture right here. We have ammonia, okay, NH3, and look, there's a lone pair on nitrogen. That we said that earlier. Nitrogen always has a lone pair unless it's an ammonium ion, and it's got 4 bonds with a plus charge. So we got water here. We're going to label these. One's the acid, one's the base. So one can give a hydrogen away, one can accept a hydrogen. How do we know which one? They both have hydrogens. Well, ammonia here is going to act as you got to look at the products too sometimes. If you don't have the products, then you have to do some more work in figuring this out. But this is the base. Why? Because it takes a hydrogen. This lone pair grabs a hydrogen off of water. So water is acting as the acid. Okay, now what do we have over here? We have the ammonium ion, and then we have hydroxide. That's left over from the acid. See how one hydrogen was given from water to ammonia, okay? All right, so that's exactly what we just showed is what is said right here in bullet point 2. Amines act as bases because the lone pair on the nitrogen accepts or forms bonds with H plus ions. Okay. So that's one thing that I want you to keep in your back pocket for just a minute. That's one property of these amines. Now we're going to address hydrogen bonding, and then we're going to look at how these two things affect the overall properties. So hydrogen bonding, we're always concerned if molecules can hydrogen or bond or not. That's why I had you do a whole lab on looking at all the different functional groups and determining if things can hydrogen bond. Okay, hydrogen bonding can happen 2 ways, with primary or secondary amines. I'll show you those in a second, but they can only happen 1 way with tertiary amines. Okay, so look down here. We've got a secondary amine, and it's got hydrogen bonds with water. So look right here. Why is it secondary? Here's the nitrogen, and it's got 1 bond to carbon and another bond to carbon. Don't worry about the dotted lines and the wedges. The dotted line just means it's pointing back out of the plane of the paper behind us, and the wedge means that carbon group is pointing towards us. Okay, so we see this nitrogen also has a bond to hydrogen. And then that hydrogen is forming right here, the dotted line, this is the hydrogen bond with this water molecule that's highlighted in red. Okay, so the hydrogen here is partial positive, and we know the oxygen is partial negative, so you get a hydrogen bond between those two. So you see that hydrogen bonds can happen with any hydrogens that are bonded to the amine nitrogen. Hydrogen, I'm going to say that again. Hydrogen bonds can occur with and through this hydrogen that is bonded to the nitrogen on an amine group, because that hydrogen is positively polarized. Okay, it's a donor. You can also get a hydrogen bond directly with the nitrogen. So look over here at this next one on the right. A secondary amine. We've got a lone pair on this nitrogen. So that's where the partial negative comes in. And this time we have a hydrogen off of a water molecule. The water molecule is highlighted in pink. And that hydrogen is positively polarized. So that's where this hydrogen bond, this dotted red line, comes in. So there's two ways you can get a hydrogen bond. You can hydrogen bond through the lone pair on the nitrogen, or you can hydrogen bond through the positively polarized hydrogen that is bonded to the nitrogen. That's what I showed you in the first one. So that's two ways it can hydrogen bond. Okay. You can also hydrogen bond between two amine groups. So this is an amine group, and this is another amine group. The lone pair on the nitrogen of one amine group can hydrogen bond with this hydrogen on the next door neighbor amine group, because that hydrogen is positively polarized, and we know the lone pair on the nitrogen is negatively polarized. Erase all this so you can see that more clearly. Okay. So you can hydrogen bond, this can hydrogen bond with water, it can hydrogen bond with other amines, as long as there is a hydrogen on the amine group. The reason that tertiary amines can only hydrogen bond with water and not themselves, so look over here. Here I'm explicitly showing you the difference between primary and secondary amines and tertiary amines. Primary and secondary amines have some kind of hydrogen bonded to the nitrogen. And these hydrogens are always positively polarized because nitrogen's pretty electronegative. But a tertiary amine has no hydrogens, it's all carbons. Okay? It could be 3 different carbon groups, it could be 3 identical carbon groups. But we have no hydrogen here. So you can hydrogen bond with water. Okay, so the hydrogen off of a water molecule would be attracted to that electron pair on the nitrogen, but that's the only way it can hydrogen bond. There's a lot more options with primary and secondary amines. Okay, and I'm just talking about hydrogen bonding here. Okay, any questions on how that happens? That is what affects boiling point. Okay, so due to their ability to hydrogen bond, amines have higher boiling point than alkanes of similar size. We've said that with almost every molecule that has an electronegative group. So alcohols, aldehydes, ketones. Once you get the ability to hydrogen bond, there's a whole lot more intermolecular forces holding the molecules together in a substance. And it takes a lot more energy to break them apart and vaporize them. Now, compared to alcohols, amines have lower boiling point. So if I ask you to put these 3 groups in order, like if I gave you a picture of an alkane or an alkene, and then like an amine and an alcohol, this would be a great exam question. Look at the boiling points here. They all have similar size, so their molecular weights are all very similar. That's what MW stands for. So we got butane. This is just an alkane. This cannot hydrogen bond. No hydrogen bonding. The amine can hydrogen bond, but notice that the boiling point is 48 degrees compared to propanol, which has a boiling point of 97 degrees. That's quite a difference. Both of these can hydrogen bond. So why are the boiling points so different if the molecules are same size? Okay, well, amine hydrogen bonds are weaker than the hydrogen bonds that alcohol form. Okay, alcohol has OH. That looks just like water. Water is HOH. It means there's something about it, okay? The nitrogens are not quite as electronegative as the hydrogen. So it's just a stronger polarizing separation of charge. And that's why the hydrogen bonds are stronger for an alcohol than they are with an amine. That's why the boiling points of alcohols will always be higher than the boiling points of amines, okay? Questions on that? Lastly, solubility. Recall, always remember this phrase, like dissolves like. So if an amine is polar or largely polar, then it will be soluble in water. But if it's mostly nonpolar, then it will not be soluble in water. So just like alcohols, remember when I told you the carbon-hydrogen ratio? In fact, that was a test question. I said, why are some alcohols soluble in water? Why are some not? It all has to do with the fact that you've got both a nonpolar portion of the molecule and a polar portion of the molecule. So just like alcohol, same idea here. The greater the carbon content of the amine, the lower the solubility of the amine in water. So what that means is if you have an amine that looks like this, NH2, and then you've got this long hydrocarbon chain, okay, like 6, 7, 8 carbons in it. This one is not going to be as soluble in water, or it may not be soluble at all, compared to an amine that looks like this. Just 1 carbon, or 2 carbons, or 3 carbons. That makes sense? This would be water soluble. Okay, you could look at the nitrogen carbon ratio if that helps you, just like we looked at the oxygen carbon ratio for alcohols. Okay. Okay. Let's look at these 3 examples. Arrange the following compounds in order of increasing boiling point. Explain why you place them in that order. So A, B, and C. Did I have a poll question for this? I don't think I did. Yeah, no, I didn't. What do y'all think? Which one would have, it says increasing boiling point. So which one would have the lowest boiling point? A, B, or C?
B.
B. Robin says B. Anybody agree or disagree? Here's what I noticed about B. You have is it a primary, secondary, or tertiary amine?
Tertiary.
It's tertiary. And how many carbons are on here?
3.
1, 2, 3, 4. So can this one hydrogen bond with other tertiary amine groups? It can hydrogen bond with water, but can it hydrogen bond with other tertiary amine groups?
No.
Like if you just had a test tube with the pure substance in B in it, you could have no hydrogen bonding. Because you have lone pair, you have an acceptor, but you don't have any donors on this molecule. None of these hydrogens are positively polarized. So you cannot get any hydrogen bonding because you have to have both a donor and an acceptor to hydrogen bond. Okay, so this one cannot hydrogen bond with itself. It could hydrogen bond with water. Everybody understand what that means? Okay. So I agree with Robin. This one will have the lowest boiling point. Because if you look at these other 2, if you look at A, we know this can hydrogen bond, and the carbon content is the same. There's 4 carbons on all of these. So we're not judging them based on how many carbons are involved here. We're simply looking at the electronegative groups and saying, can they hydrogen bond or can they not? This one can hydrogen bond, and it's OH. This one also can hydrogen bond. But we said just in the slide before, the hydrogen bonds for alcohols, so A here is an alcohol, are always stronger. So this one will have the highest boiling point, and this one will have the middle boiling point. Okay, so if we're going to put these in order, it would be B, and then C, and then A would have the highest. Questions on that?
Can C hydrogen bond with water and with
I'm
its
sorry.
Yes, Madison. Maggie, what was your question?
I was gonna say, could you explain again why? I don't want to say that whole thing over, but
Real quick, I can.
why is it in that order again?
Yeah. So basically, Maggie, B, let's look at B. B is a tertiary amine. It has no hydrogen that is directly bonded to the nitrogen. Doesn't have any. All the bonds are to carbon. You see that? So B can hydrogen bond with water. But if we're just looking at a test tube of the pure substance, like all we have is these molecules, we don't get any hydrogen bonds between the molecules. All we would have is London dispersion interactions. Okay? Because all we have here is an acceptor. No, it doesn't have no donors. Okay? Remember, a donor is a positively polarized hydrogen. We don't have any donors. To have a hydrogen bond, you have to have both an acceptor and a donor on the same molecule. So no hydrogen bonding. So the types of intermolecular forces holding these molecules together are pretty weak. London dispersion forces, or maybe dipole, but neither of those are as strong as hydrogen bonding. C and A can both hydrogen bond because you have, look, here's the donor, and then lone pairs on these oxygen, here's the acceptor on the same molecule. None of these hydrogens are positively polarized. None of those are donors. Down here, same idea. We got one lone pair on this nitrogen, and then these hydrogens, because they're bonded to an electronegative atom, such as nitrogen or oxygen, these are positively polarized. So we've got the donor and we've got the acceptor on the same molecule. That is why they can hydrogen bond with themselves and with water, but we're not looking at hydrogen bonds with water right here. So that is why we said, okay, the two that can hydrogen bond are going to have higher boiling points than molecule B, but just above on this slide, we said amine hydrogen bonds are always weaker than alcohol hydrogen bonds. So that means the alcohol hydrogen bonding is going to be the strongest. So we said it would have the highest boiling point. Does that help?
Thank you.
Okay, good. All right, moving on. Let's draw the structures of ethylamine and triethylamine. Use dashed lines to show how they would form hydrogen bonds to water molecules. Okay, so I'm just going to do ethylamine for time's sake. It's already 10:06. Oh my goodness. Ethylamine. So we have nitrogen, and we just have one ethyl group, right? This is not diethylamine or triethylamine. It's just one ethyl group. So I've got CH2, CH3, and then these other two bonds on nitrogen have to be to what?
Hydrogen.
Yes, Claire, hydrogen. So there is ethylamine. Then I can get hydrogen bonds two ways. So I can have the hydrogen on a water molecule. Actually, I'm gonna do it like this. There's my water molecule. I could get a hydrogen bond. I'm gonna draw a dashed line. I don't know if y'all can see that red very well. Between the hydrogen and the lone pair, that's one hydrogen bond. And then I got lone pairs on this oxygen of the water molecule, and it is attracted to this positively polarized hydrogen on my amine. So I get a hydrogen bond there. You could also get hydrogen bonds down here with this other positively polarized hydrogen, but I'm not going to draw that out right now. So this is very similar to what you've done in the hydrogen bonding lab. Questions on that? Okay, heterocyclic nitrogen compound. This slide is going to be kind of quick. I just wanted to show you nitrogen can be a part of a ring itself. And we have to acknowledge that now the ring is not just carbon and hydrogen anymore, that there is some kind of different atom involved. So we call these, we have a specific name for them, we call those heterocycles. Hetero meaning there's some kind of different atom here. So it's a ring that contains a nitrogen or some other atom. It could be oxygen in addition to the carbon. This is really, really important for a lot of biological molecules, okay? DNA and ATP both have this molecule here known as adenine. So adenine triphosphate, that is ATP. Adenine is part of the backbone of DNA, or one of the base pairs, sorry. And so you see all these nitrogens in this ring structure. The only one I want you to point out and know is imidazole here, okay? I want you to be aware of these other ones, but the one I want you to know, like you could draw out its structure if you had to, is imidazole, because this is in histamine, and this is exactly what Madison was talking about earlier. There are nitrogen groups in things like proteins. Histidine is a prime example. This is part of the side chain of the amino acid histamine. So we're going to get to that later when we talk about proteins, but I'm introducing this to you now, one, because it's an example of a heterocycle, and we're talking about amines, but this is going to matter a whole lot when we get to proteins. So know imidazole, be able to draw. If I ask you on the exam, what is imidazole? Draw the structure of imidazole. I would want you to know what that means and be able to draw it out, okay? It is a 5-member ring, 3 carbons, 2 nitrogens, and then one of those nitrogens is bonded to the H group. And take note of where the double bonds occur. The double bond occurs here, okay, between these 2 carbons, and it occurs here between this carbon and the opposite nitrogen, the one that doesn't have the H bond, okay? All right, that's all I'm going to say about that slide. I have a poll question. I just want you to tell me which ones are true heterocycles and which ones are not. Okay, you can select multiple here. A, B, C, and D. Just select the ones that are true heterocycles. Okay, 6 out of 7. Let's get the last vote in. All right, in the poll. Okay, share results. So everybody said A. 7 out of 7 said A. So I agree. We've got this nitrogen, and I know it looks hydrogen atom in the ring, but it's not. The nitrogen is the one that's in the ring. We know hydrogen cannot have 2 bonds. Okay, so that looks deceptive. I don't like the way they drew this actually. If I was drawing this ring, I would have drawn nitrogen bond to hydrogen out here. Okay, but this nitrogen is in the ring like this. Okay, so there's that one, and then there's all your carbons. B is not a heterocycle because this nitrogen is not a member of the ring. If it was where the arrow was pointing to, it would be, but this one's not. This is aniline. Okay, nobody said C, and I agree, this one's not. D is, yes, you got this nitrogen right here, part of the ring. It's got to be inside the ring. Okay, so A and D are the correct choices there. Okay, the last part of this lecture today is going to really focus in on what we mentioned earlier about how amines can act as bases. So, amines, we saw this already, but they can react with water to form ammonium ions and hydroxide. Or you could write OH minus, okay? Hydroxide is a polyatomic that you got to have memorized. So here's 3 examples. We all have these different amines. We got a primary. This one is a secondary, and this one down here is a tertiary. All of them can react with water because all of these have lone pairs. Okay, it doesn't matter how many hydrogens or carbons are bonded here. If the nitrogen has a lone pair on it, it can pull a hydrogen ion off of an acid. Okay, water here is acting like the acid. These are all acting like the base. Okay, and then on the product side, here's what you see. You see this plus charge associated, that is on the nitrogen. So if you were to draw the Lewis structure out, you would see 4 bonds to nitrogen. Each of them have a plus charge, okay? And then you're left over with the hydroxide. That's what's left over from water once it gives a hydrogen to the amine group, okay? What I want you to notice is that all of these reactions are, look, what does the double arrow mean? All the reactions are what?
Equilibrium.
They
Or
can
they
have
convert?
equilibrium because they can go back and forth. What's the word we use to say they can go back and forth? Reversible. Okay, which, yes, Madison, means they can all reach equilibrium. Okay, so because they're reversible, look, if we're looking at it from the other angle, if we have an some kind of NH4 molecule or something that has a plus charge on the nitrogen, ammonium ion, plus we have OH minus. Now, which one is the acid and which one is the base? What do y'all think? Do y'all remember anything about OH minus and what kind of base it is? OH minus is a really, really, really strong base. It's like the strongest base you can have. So now guess what? This is the base. And this, our amine compound, but it's an ammonium ion here, becomes the acid. Okay? And then you just run the reaction backwards. Okay, you run it backwards to see how the amine group acts as the acid. So what we're saying is that ammonium ions, right here, they can react as acids in the presence of a base. And you regenerate the amine, but there has to be a nitrogen bonded to that hydrogen group. Okay, so what I'm saying is that if you have a nitrogen that looks like this, it's bonded to 4 carbon groups and it's got a plus charge. Just because you have a nitrogen with a plus charge on it, if it's a quaternary amine, that's not going to act as an acid. As long as there is a hydrogen bonded, so you got a primary, secondary, or a tertiary amine, that can act as an acid and it could give the hydrogen away. So quaternary amines cannot act like acids. And I think I have a slide on that in a second. What do these other bullet points say? Ammonium ions are also formed when amines react with H3O+. Okay, so H3O+ is kind of interchangeable with H+, but that's another form of the acid. To name the products here, we simply replace the amine. Instead of ending it with amine, we call it ammonium. Okay, so for example, let's say you had something like this. What are my products going to be? Let's get this over. My product, since this is the acid and this is going to be the base, this nitrogen right here is going to grab the hydrogen off HCl and it's going to form NH4+. That plus charge is now in the nitrogen. And then we've got Cl- left over. So how do we name this? We would name it ammonium chloride. Okay, we would just, instead of ammonia, now we call it ammonium. And then this would actually be an ionic compound. Okay, cation and anion. So we call it ammonium chloride. We're going to do more practice with naming these ammonium salts in a minute, if we get to it, hopefully. I think a really important point on this slide is right here, this last bullet point. At physiological pH in blood and body fluids, amine compounds exist as ammonium ions. So at a neutral pH, the amines are actually in their charged forms if they have the option to be. So for example, a really great example, histamine, side chain on a protein. Those nitrogens carry a plus charge. Or that one in particular. Serotonin, which is a neurotransmitter. You see the plus charge on this nitrogen over here. Okay. I can get to it later when we get to proteins about maybe why some nitrogens won't carry plus charges and others will. I'm not going to split hairs and ask you about that on exam 2. Like which nitrogen would carry the plus charge, but we will get to that later. Okay. But you notice that some nitrogens in body fluids carry plus charges. Okay, write the balanced equation for the reaction of ammonia with water and for the reaction of ethylamine with water. Let's not do ammonia with water. Let's just do ethylamine with water. You could practice with ammonia and water on your own. It would look a whole lot like what we drew up here, except for instead of HCl, it'd be H2O. So we're going to draw a reaction of ethylamine with water and label each species in your equation as either the acid or the base. So ethylamine, you've already drawn ethylamine, I think. We drew that earlier. That is a nitrogen. And we've got this ethyl group, CH2, and then CH3. And then we got 2 hydrogens branched off the nitrogen. There's ethylamine, and we got water. Okay, so which one is going to be the acid and which one's going to be the base?
Would H2O be the acid?
Yes. Thank you, Maggie. And so that means the amine here is going to be the base. Okay, so that means this nitrogen group is going to grab a proton or a hydrogen ion off of water. So draw the product you would get. So everything about this amine is going to be the same except for what? We're going to have CH2, CH3, then we got the hydrogen, and guess what? Now up here, instead of a lone pair, this is a bond to hydrogen. Now, is there anything else we need to put on this molecule or are we done?
plus in the nitrogen?
Yes, Maggie, good. A lot of people will forget that you've got to now put a plus charge. It was neutral, it gained a hydrogen ion, so now it has a plus charge. And what's left over from the water? It gave a hydrogen away, so all that's left over is an O and an H, and it has a negative charge now. So don't forget to acknowledge how the charges change. If you go from neutral and you gain a hydrogen ion, you become plus charged. If you're neutral and you give away a hydrogen ion, you become negatively charged.
Could you explain how we knew the ethylamine was a base?
Yes, so this actually really gets into like acid-base strength, which was all part of chapter 10. So if you want a quick review on that, Claire, I don't have the textbook sitting right here by me, but if you have your textbook, somebody remind me, I will try to send you the table of acid-base strength. So this is all kind of inherent knowledge from a previous course. In a previous chemistry course, you should have covered acid-base strength, but water acts as either an acid or a base depending on who it's around. Okay, so if it is in the presence of a molecule that has a lone pair on it, oftentimes water will act as an acid. Okay, this lone pair is really electronegative, we know that, and it's really attracted to these hydrogens, and it will pull one off. Okay. But if water is in the presence of a strong acid, so if you have something like this is a strong acid. Okay, it's one of the 6 strongest acids. Now, yes, there are lone pairs all over chloride here, but that doesn't make it a base. Okay, and this is where acids and bases can get kind of confusing. Hydrochloric acid is a very strong acid. So here, water's in the presence of a strong acid. So water, these lone pairs on oxygen, are going to act like a base and pull this hydrogen off of chlorine. So water is not always an acid, it's not always a base. It depends on who it's in the presence of.
Okay, that makes sense. It's coming back to me.
Okay.
Thank you.
Yeah. Okay. We have like 7 minutes left. Hold on. We got some practice. Let me see what we have left. We've got some practice problems here. We might have to finish this another time, but let's go ahead and do this histamine practice problem and do one of these, and then we're going to spend just 1 minute talking about quaternary amines, and then we're going to be done. Okay. So this histamine practice problem, it says histamine, it's an amino acid, will react with acids such as acetic acid to form ammonium salts, okay, which themselves are weak acids. Now, when treated with KOH, so this is a really strong base. OH minus, anything with OH minus in it is a strong base. So ammonium hydroxide, lithium hydroxide, potassium hydroxide, all strong bases. Okay, so the free amine will be regenerated. Write a balanced equation for the reaction of histamine acetate with potassium hydroxide. What in the world? This sounds like a lot. So let's break this down. Here's what I see. Plus charge, minus charge. This is histamine acetate. Now, you may not recognize acetate, but it is one of the polyatomic ions that I always ask my students to memorize in our previous chemistry class, okay? So this is an ionic compound. You just see it written as 2 different ions here, the positive and the negative, the cation and the anion. Okay, so that's histamine acetate. Here's potassium hydroxide. I want you to think of this as a double displacement reaction. The cations are going to switch, and the anions are going to switch, kind of, sort of, okay? What it said when treated with KOH, the free amine is regenerated. So this is not going to be a full double displacement reaction. Here's what's going to happen. This is going to act as the acid. This is going to act as the base, okay? And then we know if we were to break potassium hydroxide up, we get K, here's the cation, and here's the anion, KOH, okay? So if we're going to regenerate the free amine, what does that mean? Free amine. I'm going to call this part of the molecule H for histamine. Okay, so I'm not going to draw it all out over here. I'm just going to say we got histamine, and that histamine is bonded. Here's our amine group. On the reactant side, we have NH3. So there are. If we look at this nitrogen, if we draw out this nitrogen, we've got 1, 2, 3 bonds to hydrogen. There's the 3 hydrogens, and then we have this bond to carbon, which leads to the rest of the molecule. So that's why I got the plus charge here. The free amine, though, will look like this. And it's confusing that I wrote another H over here for histamine, so I'm actually going to say HIST, histamine. Okay, this is the rest of the histamine molecule. So this is the free amine. Okay, so how did we get that? Here's what happens. These lone pairs on this OH group, remember we said OH minus is a really strong base? It grabbed one of these hydrogens off. So the amine group here is acting like an acid because it's giving a hydrogen away. We nickname acids as proton donors. They give away a hydrogen. And that's what's happening here. One of these hydrogens is going and it's going to go off and it's going to bind with OH minus. So what does that make? That makes one of our other products is water, HOH. Okay, so that's how this happened. Well, what about the acetate and the potassium and all that? These are spectator ions in the reaction. Do you guys remember spectator ions? Okay, think of like a spectator at a ball game. Are they in the game? Are they in the reaction? No, they're just watching. So they're just floating around over here. You could write them as potassium C2H3O2 if you wanted. This is potassium acetate. Okay. The histamine and the OH minus, these are the products you get from that. Those are the ones involved in the reaction. That's the acid and that's the base. The acetate and the potassium are not involved. Okay. Okay. I want you to do this first practice and then we're going to be done. We're going to take a break. So just do A here. Draw me the products. Which one's the acid and which one's the base? So you should have that the amine is the base here. And HBr, this is hydrobromic acid. So what's going to happen is the lone pair is going to grab this hydrogen from the acid, form a bond. Okay, who's got the products? What is this, by the way? This is isopropyl N-methyl isopropylamine. So now we're going to have the same, this is our isopropyl group. This doesn't change. And there's the hydrogen on that carbon. And then this is the nitrogen. There's the methyl group. And now we get this hydrogen. I'm going to draw it in black, actually. This new hydrogen bonded up here. That was from HBr. And what else should we draw on this amine compound now? Aggie said it earlier.
It should be positive now, shouldn't it?
Uh-huh. And what's our other product?
BR minus.
Yes. Okay. So if I give you an amine reaction on an exam, if the nitrogen has a lone pair on it, it's going to act like the base. It's going to accept the hydrogen. The only one that's different is down here, D. We don't have time to get into this because I'm done. But here, the nitrogen has 4 bonds, it's got a plus charge, and it's in the presence of a strong base. So this one will be the acid, and this one will be the base. Okay, so the way this product or reaction will work out is opposite of what we just saw. Okay. All right. I think there's only 1, 2, 3 slides left. When we come back, we'll finish these real quick, and then we're going to go over exam 1. So I'm going to take a break. It's 10:30. Let's say 11:05. Okay, let's take a 35-minute break. We'll get back on at 11:05. That'll give me time to get up to